Okay, for the study guide, let's go through the questions that were covered in the recording of slides 1 through 16 for chapter 12. So question number 1 says list 3 important properties a substance must possess in order to serve as the genetic material. So one of those was that it has to be able to store code or store genetic information, I guess you could say, in the form of our genetic code. And also it has to be stable, has to be a stable molecule and able to replicate accurately. And then what was the other one? Gosh, I can't think of it. It must, let's just look it up because I can't think of what it is. Must be able to undergo mutations so that we can have genetic variability. Okay. Must be able to undergo mutations. Okay, sorry about that. And then on the next question, it says transformation of bacteria was shown to occur when blank bacteria were injected into mice. So the transformation came from 2 types of bacteria. One was dead S strain, or we actually should say heat-killed S strain bacteria, because live S strain killed the mice, but heat-killed S strain did not. So the heat-killed S strain mixed with live R strain. Those bacteria were injected into mice and caused the mice to die. So the mice died. That goes in the second blank. And that is when we knew that there must have been some sort of transforming substance that left the S strain and entered into the live R strain and gave it the characteristics of being deadly and caused it to produce a capsule. So number 3, DNA was shown to be the transforming substance when only the blank enzymes could inhibit transformation. So in the experiment by Avery, that enzyme was called DNA ACE. And I know that in the slides, the big A is not there, but I'm pretty sure it is there in the actual homework questions. So I mean, it's not like it would keep you from getting the answer right on the exam, but I'm pretty sure that we should say DNA ACE instead of DN ACE. So, number 4, before discovering that DNA was the genetic material, why did some scientists believe DNA could not be the substance which contained our genetic code? And the answer is because DNA only had 4 types of nucleotides. Which we know serve as the building blocks for DNA. So there were only 4. And the next question kind of continues with that whole idea. Before discovering that DNA was the genetic material, why did some scientists believe protein could be the substance which contained our genetic code instead of DNA? So DNA only had 4 types of nucleotides or building blocks, but protein had 20 types of amino acids or building blocks. So it seemed that proteins could hold more genetic information. Number 6, besides DNA and protein, which other substance was considered as the genetic material? And that was RNA, which we will learn more about in this chapter. Number 7, why was the X-ray diffraction photography of Rosalind Franklin and Maurice Wilkins considered critical evidence in the study of DNA? And it clearly showed that DNA was shaped like a double helix or twisted ladder. Number 8, in the Watson and Crick model of DNA, the steps of the ladder are composed of a purine and a pyrimidine base. And the 3 components of a nucleotide are a phosphate group, the sugar, so we'll say deoxyribose sugar, and a nitrogenous base. Which 2 nucleotides are purines? They're the double ring structures, and I'm just going to use the letters. So the 2 that are purines are A and G, and they have a double ring structure. That's the answer to the second question. Which 3 nucleotides, and this time we have to include one nucleotide that's only found in RNA, but the pyrimidines are cytosine, thymine, and uracil is the one that's only found in RNA. And pyrimidines only have a single ring structure. Number 12, an unknown chemical is analyzed and found to contain the bases thymine and guanine. So it could be RNA because of the guanine, but thymine means that it has to be DNA. RNA would have uracil instead of thymine. So the chemical is most likely DNA. Summarize Chargaff's rule, and Chargaff's rule just stated that the amount of A in any sample of DNA from any species, the amount of A is equal to the amount of T, and the amount of C is equal to the amount of G. And we use that now in our rule of complementary base pairing to say that A's always pair with T's and C's always pair with G's in the double-stranded DNA molecule. Now, we've got a math problem. So the DNA sample contains 13% adenine. So we already know thymine is going to be 13%. Thymine is 13% because it will be the same as adenine. But what about cytosine and guanine? Okay, so what we have to do is remember that percents add up to 100. So we have to do a little math. We have to say 13 plus 13 is 26. So what is 100? Take away 26. And that is going to be equal to 74. So the cytosine and guanine together make up 74%. So then we have to figure out what each one is. And since they're equal, 74 divided by 2 equals 37. So cytosine is going to be 37%, and guanine is also going to be 37%. And just to check, make sure they all add up. 37, 37, 13, and 13 add up to 100. So that is how you would figure it out. As long as you're given the percentage of one of the nucleotides or one of the bases, you can figure out the other 3. Now, starting with number 15, that's going to be, we're going to be talking about DNA replication. So let's move on to the PowerPoint and go to the section on DNA replication. Okay, so basically DNA replication is when you have, from current slide, one strand of DNA that copies itself, and that happens during interphase, and it's during the S stage of interphase. So we have, I like to teach DNA replication, well, all of the processes, replication, transcription, and translation, based on the enzymes that are involved, because the enzymes kind of tell you what, their name tells you what they do. So the first thing that has to happen is, since DNA is a double-stranded molecule, it has to unwind or separate. And the shape of DNA is a helix. So the enzyme that unwinds and separates the 2 strands of DNA is called DNA helicase. So DNA helicase first unwinds and separates the 2 parental strands of DNA. And then single-stranded binding proteins attach to prevent the helix from reforming. The next thing that happens is the replication process starts, and it begins with DNA primase. DNA primase is an enzyme that places short primers, which are short strands of short sequences of RNA, on the parental DNA. And then DNA polymerase recognizes the RNA primers and starts adding nucleotides to the old or the parental DNA strands. And this is what it looks like. So first you have DNA polymerase comes in and adds nucleotides continuously to the strand we call the leading strand. This is the helicase. DNA polymerase is this brown molecule that looks kind of like a big fat half moon or crescent moon, but it's just a fat version of a moon. But it replicates the leading strand by adding nucleotides that are complementary to the parental strand. And it just adds them continuously, adds them, one after another until that piece of DNA is replicated. But the other strand, which is called the lagging strand, is replicated in pieces. It's replicated in little pieces of DNA called Okazaki fragments. So you have your helicase that unwound the parental strands. You have your single-stranded binding proteins that keep the parental strands from reattaching to each other. And then you have DNA polymerase that comes along and finds the RNA primer, which is represented here by this little purple piece. That's actually RNA nucleotides that are attached to the parental DNA. And they also attach by complementary base pairing. So the DNA polymerase then adds nucleotides to that little piece called an Okazaki fragment. Then it comes in and adds nucleotides to the other pieces. And after that, in the lagging strand only, an enzyme called DNA ligase seals the fragments together to form one strand, one continuous strand. So what you end up with in replication, in DNA replication, is you end up with, the process is called semi-conservative. You end up with two new strands. Let's blow this up so you can really see it. Two new strands of DNA, and each strand has one old strand from the parental strand, and then one new strand that was added by DNA polymerase. And if we know the sequence of the parental strand, then we know the sequence of the new strand because it's complementary. So every time there's an A, then it will pair with a T. If there's a T, it will pair with an A. C will pair with G, and G will pair with C. So we can always know, if we know the sequence of the parental strand, we can always know the sequence of the new strand that's complementary to it. So we say that DNA replication is semi-conservative. Make sure you know that word, semi-conservative, because each new DNA strand has an original strand and a new strand in it. Okay, as far as the accuracy of replication, DNA polymerase is very accurate, but it still makes mistakes. It makes, and these are questions that are in your homework, and so they might be on the exam. So the mistakes that are made during replication are about one mistake for every 100,000 base pairs. However, DNA polymerase comes in and can identify and correct most of those errors. So the actual mistakes are that end up still being mistakes are about one in every let me look it up. I think it's million, but it might be billion. Hold on. The actual mistakes that get made after DNA polymerase makes its corrections are, it's 1 in 100 million. In fact, let's see if I can write that in here. 1 in 100 million. 100. Ugh. This is why I don't like to do this, because that always happens and I don't know how to stop it. So the mistakes that get made by DNA polymerase are one mistake in every 100,000 base pairs. But after DNA polymerase comes in and corrects those mistakes, there still are 100 million, one mistake in every 100 million base pairs. And our entire DNA sequence for our entire genome or genetic code is billions. We have billions of base pairs. So 1 in every 100 million mistakes are made, and we're going to learn that some of those mistakes, even some of those mistakes, don't lead to problems in the actual protein that's produced. So we're going to stop here. This is slide number 25. We're going to stop with slide 25 and answer the questions on DNA replication, and then finish our transcription and